Hey everyone, my name is Kayla Bramlett and I'm a rising senior biochemistry major. Under the guidance of Dr. Swain, my research this summer was looking at glucose transporter proteins in Saccharomyces cerevisiae, specifically HXT2, and how its function relates to its structure. Just to give a brief introduction, I'm going to start by telling you about glucose transport, why we're using Saccharomyces cerevisiae, or yeast, as a model, and a little bit about my protein. Glucose is the main carbon source for most eukaryotic cells. In yeast, glucose enters the cell through facilitated diffusion in the plasma membrane. It does so by using hexose transporter proteins, or HXTs. This is seen in Figure 1. Depending on the amount of extracellular glucose present, different transporters are mobilized to the membrane. However, the question of how the amount of glucose present is sensed and signaled remains unknown. This is what my research attempts to answer. So why yeast? The answer to this is actually pretty simple. Yeast shares many similar components and pathways for glucose sensing and signaling with its mammalian counterparts. Yeast HXTs are also homologous to human glucose transporter proteins, also known as GLUTs, making it a good model system. Yeast has 20 known hexose transporter proteins. These proteins have a 12 transmembrane domain with the N-C termini located in the cytosol, where they are able to move freely. Yeast is able to tolerate a wide variety of glucose levels because glucose transport occurs by two distinct systems, one that has a high affinity for glucose and one that has a low affinity for glucose. The four main proteins we normally look at are HXT1, HXT2, HXT3, and HXT4. My protein of interest for this study, however, is HXT2. HXT2 is considered a high affinity transporter. This means it is expressed under low levels of glucose. HXT4 is also a high affinity transporter, while HXT1 and 3 are low affinity transporters that are expressed under high levels of glucose. Now I'm just going to give you a quick overview of the preliminary data that helped lead to my hypothesis. What we are seeing here are HXT overexpressors. Overexpressors simply mean the gene is continuously expressed, and therefore the protein is always present in the membrane. What Figure 2 is showing us is that when HXT2 is overexpressed, it is slightly toxic on low glucose. The first thing that is interesting about this is that, as stated earlier, HXT2 is normally expressed under low levels of glucose. So why, when it's overexpressed, do we see slight toxicity? The second thing that raises suspicion is that HXT4 overexpressor is completely toxic on low glucose. This is also interesting because, like HXT2, HXT4 is a high affinity transporter expressed in low glucose. So why, if both HXT2 and 4 are normally expressed under low levels of glucose, then why, when they're overexpressed, do we see these differences? Next, we thought, well, okay, since we see these differences in the overexpressors, maybe this can be explained by deleting different endogenous transporters. Endogenous transporters are the ones whose expression is under the control of its promoter, aka the glucose concentration. Again, however, we see variations in the data. While each overexpressor delete combination for HXT1 and 4 show varying phenotypes from the wild type, HXT2 shows the same phenotype all the way around. This is depicted in Table 1. Again, interesting because although HXT2 and 4 have similar functions, it seems that HXT1 and HXT4 are the ones that are lining up better. This led us to compare the sequences of these proteins, thinking there could be a difference in their structure. A tea-coffee multi-sequence alignment between the first 4 HXTs was done. What we found is that the transmembrane region of HXT1 through 4 are highly conserved. The only place we get any real variation at is at the N and C termini. So, based on all the variations we see in the overexpressors and the overexpressor delete combinations, and because these proteins are highly conserved everywhere except the N and C termini, it is hypothesized that there must be some kind of interaction between the N and C termini of the protein that is responsible for signaling and sensing different levels of glucose that could explain these variations. Before I get into methods, I just want to make a quick statement that says, due to COVID-19, we had to adapt our plans for this research and take a more computational approach. So to start off, the first thing I did was 3D modeling of my protein. Because I was only interested in the N and C termini of HXT2, and for simplicity's sake, these were the only regions of the protein I modeled. To predict 3D structure, I used a variety of online homology modeling and protein threading tools, such as Primo, Robetta, Swiss Model, iTaser, Fire2, GenThreader, and CHP models. Homology modeling compares the input sequence with homologous sequences of a protein with known structure to build the 3D model off of, while protein threading utilizes fold recognition as the template rather than sequences. Once all the predicted models were built, they were input into Chimera, energy minimized, and aligned using Matchmaker. Figure 4 depicts all the models generated for the C termini on the left and for the N termini on the right for HXT2, aligned after using Matchmaker. Now I know what you're thinking, it looks like a hot mess, but that was somewhat expected because the input sequences for both were very short. The C termini was only 36 residues long and the N termini was only 49 residues. Once aligned, each model's differences was quantified by calculating this root square mean deviation, or RMSD, in Chimera. RMSD is the measure of the average distance between the atoms of superimposed proteins. The lower the score, the more similar the two are. Therefore, the models with the lowest totaled RMSD were selected. For the C termini, this was iTaser 2, and for the N termini, this was iTaser 1. These were the models that were used from here on out. Once the best model for each termini was determined, the two were then combined into one PDB file and fixed about 10 angstroms apart in Chimera using the distance tool. Although in the full protein, the two termini are actually predicted to be about 40 angstroms apart, 10 angstroms was chosen so that the two termini would be able to see each other during the simulation. Next, we went on to the actual molecular dynamics, or MD, of things. MD is a way of computationally analyzing the physical movement of atoms and molecules for a fixed period of time. The program used for this was QuickMD. QuickMD is a visual molecular dynamics, or VMD, plugin. QuickMD goes through 4 main steps during the simulation. The first is energy minimization, then annealing, then equilibration, and finally the molecular dynamics step. For the molecular dynamics step, one residue on each termini that would normally be fixed in the transmembrane region was also restrained in QuickMD. This was done to mimic how it would actually be in the cell. QuickMD is extremely user-friendly and automatically creates the necessary input files and output files as it goes. Once the simulation was done running, analysis tools in Chimera and VMD were used to look at any interactions. Using Chimera, we are able to look at what took place during the molecular dynamics simulation. This is what I'm showing you now. As you can see, although a few residues are interacting in the middle, the bulk of both termini do not seem to truly be interacting. In total, the MD simulation was 4 nanoseconds. To determine if the simulation reached a steady state equilibrium, we looked at the RMSD. Because the MD simulation started from one snapshot, it is not a good representation of that protein's real-life conformations. Using RMSD helps us track the time when the protein is equilibrated, assuming that the equilibrium conformations are the most energetically favorable and the most stable. If the simulation had reached this state, we would see a plateau, meaning all the up and down spikes seen in Figure 6 would eventually even out. Although it is not possible to achieve a perfect equilibrium, if the program runs long enough, we should start to see this plateau. As you can see here, however, we do not achieve this state. Simply, all this means is that any interactions we do see are considered transient interactions, or those that are easily formed and broken. Although a longer simulation is needed, we still looked at the interactions formed during this run. For simplicity, we only analyzed from 2 nanoseconds to 4 nanoseconds, or what is outlined in pink in Figure 6. two types of interactions we looked at were salt bridges and hydrogen bonds, which are both types of non-covalent interactions. What we see in Figure 7 is that there were 26 salt bridges formed during this time. However, only one salt bridge even occurred between the two termini. Similarly, a total of 262 hydrogen bonds were formed, but they seem to be breaking almost as quickly as they were formed. So in conclusion, it can be said that the N and C termini of HXT2 interact, but only to an extent. These interactions are not stable, and we know that they're not stable because the RMSD analysis shows that the simulation never truly reached that steady state equilibrium. This combined with the fact that there was only one salt bridge formed during the two termini during the entire simulation, and the hydrogen bonds were broken and formed extremely quickly, tell us that further analysis is needed. Overall, this is still a work in progress, and I'm currently running a longer simulation, which will hopefully be able to achieve that steady state we're looking for. Once the steady state is achieved, it is thought that we will be able to see repeating structural conformations and stronger interactions. In the future, we also hope to look at interactions between the termini of different proteins for HXT1 through HXT4. For example, perhaps taking the C termini of HXT2 and putting it with the N termini of HXT4, and so on, for a total of 16 different combinations. Lastly, I would like to thank SC Embry for funding and making this research possible. Additionally, I would like to thank Presbyterian College's Department of Chemistry and Biochemistry, specifically my advisor, Dr. Swain, for guiding me throughout my research this summer. Finally, I would like to thank Dr. Patu for taking the time to help me with the computational aspects of my research. Lastly, these are my references, and that's all I have, so thank you guys.